role of bacteria was relegated to decomposers. Okay, so all components at heterotrophic level produce detritus, either as detritus from the skin, from scales, detritus from waste, okay, or even detritus from dead organisms. Okay, so all these organisms, they have relatively short lifespan. So when they die, they form the detritus pool. And the role of bacteria at that time is known to degrade, attach to this detritus, degrade them, and then recycle the inorganic nutrients back to the phytoplankton. Okay, so inorganic nutrients such as nitrogen and phosphorus. So this is essentially the classical food chain. However, there was some problems later on with this food chain when they realized that so Pomeroy 1974 in his landmark paper suggested that the classical food chain was incomplete. Okay, essentially when they incubate seawater samples that are small, which means you take the total seawater, you filter them using a 366 micron filter. They found out that the less than 366 micron fraction is actually responsible for most of the respiration. Okay, so that means there is something missing in this classical food chain. When you filter off most of these large components, you realize that the respiration by the microbes are more significant than the macroorganisms. So if you look at this classical food chain, the transfer of biomass, the transfer of energy transfer and biomass transfer is essentially in a particulate form, okay, where the phytoplankton is eaten up by zooplankton, then the zooplankton by fish, and their waste as detritus. So this is essentially a particulate form. However, we now know that the dissolved organic matter is an important organic matter pool in the seawater. Okay. So it's nearly about half of the primary production is actually released as dissolved organic matter. So up to half of primary production is released as dissolved organic matter, and this enters the dissolved organic matter pool. Okay, so this pool is actually very large and contains most of the carbon in the seas. So you also have the contributions from microzooplankton, zooplankton, and other organisms into this dissolved organic matter pool. However, the problem is only bacteria is known to utilize this dissolved organic matter pool. Okay, so bacteria is the main utilizer of this dissolved organic matter pool. So what the bacteria does is it takes up the dissolved organic matter and incorporates it into its biomass. Okay, so If there is no bacteria, this dissolved organic matter pool remains lost outside the food web, outside the food chain. So this organic matter, this source of organic matter, source of carbon, source of energy, remains lost if there is no bacteria involved. Okay, because only bacteria can play the important role of repackaging the dissolved organic matter into bacterial biomass. So from dissolved into particulate, bacteria is able to perform that function. Then nowadays we also know that bacteria that is produced is graced by protozoa, protists, protozoa, nanoflagellates, okay, and after this, protozoa is eaten up by microzooplankton. So energy and carbon that is otherwise lost is shunted, is channeled back into the classical food chain via the microbial loop. Okay, so this is known now as microbial loop. So this term is actually coined by Azam, 1984. Okay, so he's one of the oceanograph microbial oceano mi- microbial ecologists in Scripps Institute of Oceanography. So he coined the term microbial loop, where carbon and energy from the dissolved organic matter pool is channeled back into the classical food chain via bacteria, protozoa. Okay, so without these microbes, all this energy and carbon is lost. Okay, so that's why the role of bacteria became more important when you take into account this microbial loop. Okay, so now the role of bacteria, protozoa, protists, ciliates as microzooplankton and zooplankton is important because there is regeneration of energy and biomass from an otherwise unutilizable source. Okay. However, the microbial food web is still incomplete because now we know the role of viruses, marine viruses. Okay, there are marine viruses that actually infects bacteria. Okay, and when bacteria is infected by a virus, the bacteria is lysed and goes and the cytoplasm goes back to the organic matter pool, and then dissolved organic matter pool is taken up back by bacteria. So this is actually the virus loop or dissolved organic matter loop. Okay, this is another loop. So viruses can be important up to 50% of bacterial mortality. So because once you know the importance of bacteria, you need to know the what is the fate of bacteria. Okay, you need to know what is the fate of bacteria, either being grazed by protozoa or lysed by the virus. Okay, because these two fates, okay, FATE, these two fates are different because if it is by grazing, carbon and energy goes up the food chain. However, if it is viral lysis, then the carbon goes back to the DOM pool. So the fate of this bacteria, either whether going to the protozoa or going to the virus, is important. You need to understand this in order to understand the carbon balance in an aquatic habitat. Okay. Now we also know that the virus loop is not so simple because there is lytic and also lysogenic levels. Okay, because not all bacteria that is infected will be lysed. Whether because sometimes the virus will enter the lysogenic cycle. Okay. Usually this is related to the productivity of the habitat. If an aquatic system is productive, most probably the viruses enter the lytic cycle. However, when conditions are not suitable, the virus switches to lysogenic mode. Okay, so there is also a bit of a variation in this virus loop. Okay. Now we also know that there are mixotrophs. Okay, mixotrophs are organisms that are capable of both autotrophy and heterotrophy. This is especially important in the protozoa and the microzooplankton fraction, because microzooplankton and protozoa, they have components, they have organisms that can sometimes carry out both photosynthesis and also grazing, both phototrophy and heterotrophy. So the marine microbial food web now is more complex. When you're talking about the marine microbial food web, this should be your final food web, but you have to have details. For example, the mixotrophy here. You should also have details on the lysogeny and lytic modes here. Okay, the importance of bacteria, the importance of the dissolved organic matter pool. Okay, so the classical food chain incorporated with the microbial loop, viral loop. Okay, all this becomes the marine microbial food web. Okay, you have any questions? Okay. So if there are no questions, okay. So why is it important to know this? Okay. From the first few lectures itself, we have already discussed the importance of CO2 emission towards global warming problem. Okay. More and more CO2 in the atmosphere leads to global warming, okay, and leads to climate change. And we know the importance of phytoplankton in fixing the CO2 from the atmosphere. So phytoplankton plays an important role removing the CO2 from the atmosphere. Now we know that up to half of primary production can be released into the DOM pool. So you need to know the dynamics of this DOM pool, the drivers, what is affecting the DOM pool, in order to understand how to overcome the increasing CO2 emissions in our atmosphere. Okay, because now you know now we know that there is net autotrophy and net heterotrophy in our waters. Okay, so this is a balance between the phototrophy versus heterotrophy. So in the marine microbial food web, the major components are phytoplankton, the primary producers, and bacteria, where the phytoplankton is the main phototroph, and bacteria is the main heterotroph. So the balance between these two components will determine whether the habitat that you are studying is net autotrophy, which means it becomes a sink of CO2, or net heterotrophy, where it becomes a source of CO2. For example, in North Pacific Ocean, we know it is net autotrophy, where the CO2 sinks, okay, because the primary productivity there is so high, it overwhelms the community respiration. Okay. Whereas for coastal waters, okay, especially the coastal waters in tropics, there is a tendency towards net heterotrophy, where the community respiration is higher than primary productivity. So if tropical seas are already net heterotrophic, with global warming, where the temperature increases, what will happen? Okay, will there be more? Will our waters be a bigger source of CO2, or will it change to a sink of CO2? So in the face of environmental change, how will our habitats change? How will our habitats change when the environment changes? So the two main environmental changes for our waters, as I mentioned earlier, is warming and eutrophication. So with eutrophication, will our waters become more heterotrophy or more autotrophy? With warming, will our waters become more heterotrophic or autotrophic? In order to answer these questions, you have to measure the processes here first. Only when you understand the processes here will you be able to predict how the habitat will change in the future. Okay, any questions? Okay, so this is the marine microbial food web that we now understand and accept. But this work, the amount of knowledge here did not come overnight. Okay, a lot of it is through meta-analysis. Okay, META, meta-analysis, where they compile work from different habitats, from different countries, from different regions, and they look for patterns between organisms. So the main ones, the main literature is probably by Cole et al., 1988. Okay, COLE, Cole et al. So he's one of the first to show the relationship between phytoplankton and bacteria. Okay. So the relationship between phytoplankton and bacteria is indirect. If you look at this Foodweb. However, when he compiled data from different regions, he found that when the phytoplankton is high, bacteria is high. When the phytoplankton is low, bacteria is low. Okay, so there is the term used is coupling. There's a coupling between the bacteria and the phytoplankton. And this coupling now we understand is because of this. When there is more phytoplankton, it contributes more to the DOM pool, and this stimulates bacteria. Okay, so now we understand why there is a coupling between phytoplankton and bacteria. So, but some studies show uncoupling. Uncoupling means you study one place and then you find out that your bacteria doesn't move together with the phytoplankton. But there are other reasons. Usually there is an alternate source of food, which means the bacteria doesn't depend on the food from the phytoplankton. So when that happens, there is no coupling between bacteria and phytoplankton. Okay, so this kind of correlation uses meta-analysis or cross-system analysis. So from this meta-analysis, they have found relationships between phytoplankton and bacteria, bacteria and protozoa. Okay, and also bacteria and viruses. So from this kind of analysis, we are now able to accept this marine microbial food web as fact. Okay, although it is probably there are still probably some details that are unknown. Okay, for example, we now know mixotrophy, lysogeny, and lytic, but there are still probably other pathways that are unknown. So still worth studying, still worth investigating. We've talked about the importance of respiration. Okay. Basically, when we talk about respiration, you sometimes in some papers you'll find the term bacterial growth efficiency. The main idea for bacterial growth efficiency is because most of the measurements are on bacterial production and not bacterial respiration. Okay. However, we need to estimate respiration loss in order to find the balance between phototrophy and heterotrophy. That's why there is a need to convert bacterial production into carbon consumption. Okay. So bacterial growth efficiency, the equation for bacterial growth efficiency is bacterial production over bacterial carbon demand or bacterial carbon consumption, where the bacterial carbon consumption is bacterial production plus bacterial respiration. This is a straightforward formula, essentially to convert bacterial production to bacterial carbon demand. So by assuming bacterial growth efficiency, you are able to obtain bacterial respiration from all the data of bacterial production. Okay, so if BGE, so if you have so if you are able to estimate bacterial growth efficiency and you are able to obtain bacterial production data, you will be able to calculate the carbon consumption. So from the literature, the average bacterial growth efficiency is about 20%. So from this value, you can generate a lot of respiration data. That's why bacterial growth efficiency is used in some of the studies. However, now there are more and more people measuring respiration. So the use of bacterial growth efficiency is less and less important now.